My name is Peter Ambrose, and I had the opportunity to work at the Children's Cancer Research Institute for more than 30 years in two functions. I was heading the tumor biology group, and I was co-director of the institute. Before discussing the results of this large international study, it is essential to highlight the unique features of neuroblastomas. This most frequent tumor of infancy and early childhood demonstrates extraordinary clinical and biological heterogeneity. Approximately 60% of infants and young children suffer from very aggressive neuroblastomas in need of high-dose chemotherapy, radiation therapy, and bone marrow transplantation, and also antibody treatment. However, and this is unique to this tumor, approximately 40% of neuroblastomas show a less aggressive or even benign tumor behavior. In this group of patients, no or only mild chemotherapy is needed. The key question was and still is which markers allow a clear distinction between these different entities to allow a tailored therapy. Others and we have shown that genetic features of the tumor help to identify risk groups. To further refine prognostic markers, two international studies involving the majority of pediatric oncology centers in Europe were performed.
My name is Inge Ambrose. I worked at the tumor biology department of the Children's Cancer Research Institute together with my husband since more than 30 years. And as a MD, I brought in morphological knowledge and genetic interest, and I had the chance to combine both and use them for research purposes, but also for diagnostic purposes. The key findings, or the most important findings we got, were certainly, and this was unexpected, were certainly that the same genetic aberrations do not mean the same in different patients. And it is, of course, very important to discriminate for assessment of prognosis and for relapse probability. And we find out that the age is a very important factor which we have to include also in this tumor group of patients with localized neuroblastomas. And this means that tumor genetics, genetic aberrations of certain chromosomes, can only be assessed and precisely assessed for prognosis with the knowledge of the age.
In the last years, we have learned that tumor markers and especially genomic features are extremely helpful to learn about the risk profile of a neuroblastoma patient and consequently allow adaptation of treatment. However, in the future, even more patient characteristics need to be implemented to refine treatment approaches even further.
We would very much encourage to perform clinical studies based on the data published now, and also to include other parameters. For example, tumor metabolism, interaction with tumor stroma, interaction with the immune system. All this would be very important to include in further research projects.